Okay, very good evening, ladies and gentlemen. My name is Rino. I'm from Sunway University. So today I'm very pleasure to share with you my research topic, which is I'm investigating a specific groups of genes called PNMA family genes in modulating the pro-survival or pro-apoptotic activities in neuroblastoma SHSY5Y cells. So to be convenient, I will just say SH cells to be short form. So as a background of my study, PNMA genes is actually a very specific group of protein encoding genes in which they have conserved protein sequences. And we hypothesize that there are altered expression in SH cells, which is the focus of my focus cells of my research project. And there's one interesting thing of the PNMA genes, which is among the 15 to 19 family members of these gene groups, they have actually different functions. We call them functional divergence. And they are almost found in the chromosomes of the brain, the heart, and the human testes. So among the 15 to 19 family members, our research project singled out two main genes, which are called the MOP1 and also the ZCCHC12 gene. So we carry out the gene screening in SH cells, untreated condition, and retinoic acid treated condition. So there are two aims of my research project, which is to characterize the gene and the protein expression of the selected PNMA genes upon differentiation, and also to determine, the most importantly, to determine what are the roles of these genes in SH cells. Okay, so what have I done in my research project? Firstly, I have to make sure my cells are properly differentiated with retinoic acid. I have done a Western blot analysis as well as immunofluorescence. And also, to determine the roles, I have conducted a luciferase reporter gene assay and subsequently a knockdown assay. That means I reverse the effects of the genes. That means if they are pro-survival, I knock them down to see if they will affect the survival of the cells, and vice versa for the other effect. And eventually I will also conduct a scratch assay to see will the pro-apoptotic or pro-survival effects of the cells will affect the migration of the SH cells. Finally, I will also conduct a MTT viability assay to determine the pro-survival or pro-apoptotic effects of the cells. So our preliminary results show that these are the Western blot results. We have tested retinoic acid on the cells. That means we have shown beta-3 tubulin proteins and MAP2 proteins, which means our differentiation is okay, successful. And then, as mentioned before, the luciferase reporter gene assay, the gray color line, which is above the blue color line, above the empty vector, which shows is pro-survival for ZCCHC12. And then we have a control, and also for the MOP1. MOP1, its name suggests, is modulator of apoptosis 1, is one of the genes, and is shown it is pro-apoptotic. So yes. So in conclusion, our preliminary results show that upon differentiation, MOP1 is a pro-apoptotic gene, and ZCCHC12 is a pro-survival gene. So this can round back to what I have mentioned earlier. Among the 15 to 19 family members, the genes have different functions. So we just call that as the functional divergence of PNMA family genes. Thank you very much.
Thank you, Rino Tan. Does anyone have questions? I would like to ask a very non-scientific question. One of the names you mentioned is luciferase assay or something like that. What's the background of that name? It's a very odd name.
Oh, okay.
What's the test about? Could you explain more?
Actually, right, the luciferase assay is an enzyme. We transfect the enzyme into the cells, and then to see whether they express the luciferin That means if they express more, the gene promotes pro-survival. And then if the cells express lesser luciferin, that means it's pro-apoptotic. So it's actually to determine their roles.
I see.
Yeah.
Okay, thank you so much.